Hi everyone, welcome once again to another tutorial session in bioinformatics. In today's video, I am going to show you how you can convert Ensembl IDs to Ensembls using R. And so by the end of this tutorial, you'll be able to convert IDs such as this one here to its corresponding Ensembl, like the one I am showing now. So let's get started. For this tutorial, we are going to use this R package called biomaRt. biomaRt allows us to retrieve genomic data as well as information about that genomic data of interest from the Ensembl database. This is the Ensembl database. So what biomaRt does is that it connects to this database and then it uses this service, biomaRt. Let's just check it out. Perfect. So this service called biomaRt, and then once it connects to this service, based on the information that you specify, it will retrieve that here for you. So with the biomaRt service, you select your database and you select your data sets, and you can make your queries. But then if you have lots of gens, let's say thousands or tens of thousands of gens, then it becomes very cumbersome trying to use the browser to make queries, and that is where R comes in, or other programming environments come in handy. Okay, so when you are using programming environments, you can just write some few lines of code to process lots of gens. In our case, we want to convert IDs to Ensembl, so we can write some few lines of code to process thousands and tens of thousands of gens. So that's it. So let's proceed. So the documentation here is also extensive. I will leave the link to this page in the description box. We will just cover some aspects of it, just a few stuffs here. So first of all, you need to install your R package. I will just do that here. I have already installed it, so I will load it here. Okay, so this is my R Studio, and so I will first load the biomaRt library. So I'll say library biomaRt. That's what I'll do. Actually, let me just clear the screen. Let me just make it this way. Perfect. So I'll load it here. So I'll say library biomaRt, and then I'll load it. This is the spelling. Okay, so I'll load this library first. It's loaded now. This here, the warning messages here, you can ignore. They are just warning. I don't know why they are there, but it didn't affect the codes, and I don't think it will also affect yours if you see this coming. So just ignore the warnings. Perfect. So now we have our library loaded. Let's go back to the page. Okay, so if you want to use biomaRt to convert the Ensembl IDs to Ensembls, you need to follow the steps here. First of all, you need to identify a database. Let's just look at this one by one. This is an intro. You need to first identify your database. So with the biomaRt, because it connects to the Ensembl database, it also gives you the databases available. We have them here. Let's just do this on the terminal. The R terminal. Perfect. So let's look at it. So to get a databases, you can see let's Ensembl. This will list the databases for us. And so we have some databases here. We have 1, 2, 3, 4. Okay, so once we have the databases, we can choose the one that we want. And because we are going to convert IDs to gene symbols, we are going to use the genes database. So let's just select it. What we are doing now is exploring the biomaRt environment. So let's proceed. So we say ensemble, then we say use ensemble, and then we specify biomaRt equals genes. So what we are doing here, we are choosing the database, and we are also in the process creating our biomaRt objects. Okay, so when we are going to make queries, we need to first make sure we have our biomaRt objects, and the biomaRt objects also requires that we specify the database and the data sets. We will go through all of them. So let's start with the database. That is what we are doing now. So now we are selecting the database. So we use the use ensemble function. So we have selected it. So after getting the database, we need to also select the data sets that we are going to use. Let's go back to the browser. So this is step 1. And then step 2, we choose a data set. So let's look at how we do it. We are using the commands here. Let's just check. So let's just make a query to get the data sets available. So we say data sets, and then we say lists data sets, and then we specify the database. So here it's this object, the one we created. Okay, so with the data sets, we can check. There are lots of them. So we can just check. Let's just say data sets. This will list all of them. We have lots of them. Okay, but then we can just select a few of them. We can display a few of them. So let's say head, just to get a first 5 or 6 rows. Okay, yeah, so we have the first 5, 6 being displayed for us. You can search further. You can use patterns here. Let's just say you want to get data sets that have the name Homo sapiens, humans. We can do it here. We can search. So we can say search data sets, and then we say mart equals what? Ensemble. And then we specify pattern equals H sapiens as Homo sapiens. So it will scan through the objects here and then search for all those that have this pattern in their names. So let's run this. Okay, so we have one hit here. Take notes, this number here is not parts. Here, the data set objects is a table. Okay, so we have rows and columns. So the rows should have some form of ID. Okay, that can be used to select them. So the numbers here are used to do that. So this H1 here indicates that this row here, this data set here is row number 81. Okay, it's row number 81. So the data set, the name itself is this one here. We have name, description, and then version. Okay, so let's proceed. So once you have your data sets, you need to also select it. Now we just displayed it, so at least we have it this way. So now we know what data sets we are going to use. The ensemble IDs we are going to convert are for genes that are present in humans, and so we need to use the Homo sapiens data set. So let's look at it. And so if you are going to use a data set, you can update the ensemble objects that we created earlier, this one here. Okay, so let's proceed. At the moment, with the ensemble objects, what we have is the database. So we have to add a data set. So we say ensemble, we are updating, use data sets. And we say data set equals, and then we use this one here, this name here. So I'll just copy and paste here just to save time. Okay. And then we also need to specify the maths objects. Here it's ensemble. And so here you can change and then use any ensemble objects, any biomaRt objects. Okay, so take note of that. Here our object is called ensemble. That is why we specify that here. So let's run this. Okay, so we have our data set also selected. What you should also note is that the ensemble database is regularly updated. And so there are versions available. And so you can also select a particular version that you want to use. And so to get the archived ensemble versions, we can use this command to do it. We can say list ensemble archives. This will list the versions for us. And so you can look at the versions here and then choose the one which is appropriate for you. For this tutorial, we are going to use the 106 here. Okay, that's version 106. I think it's just, let's check. Yeah, it's this one. Yeah, that's 106. That's what we are going to use. So take note. We can also specify that. So I will show you how to specify a version in the course of this tutorial. So let's proceed. Let's go. Yeah. Okay. Let's check the documentation. Okay. So after selecting the data sets, we can make queries. That'll be the next step. So once you have your database, you have your data sets, then you can now use the getBM function to make the queries to retrieve the information you want. So let's look at that as well. Let's check. There are lots of information here, so you can read and then select the one that you see fits. Let's look at it. I'm looking at the main steps. Perfect. So after getting your biomaRt objects, you can now build the query, which is step 3. So here we use the getBM function. With the getBM function, when you call it, you need to also specify 3 arguments. We have filters, values, and then attributes. These 3 arguments. Filters and then values are used to define restrictions on the query. So it will help you to narrow down to the exact information you want, and then that information will be retrieved for you. And then the attributes, let's check. The attributes define the data that we are interested in retrieving. So with the attributes, it's the information you want. Let's say for the IDs, we have lots of information available. And so you select the one that you want, and then biomaRt will retrieve it for you. We will look at all these things in the course of the tutorial. What we are doing now is exploring. We are now exploring to try and understand the biomaRt environments. So let's proceed. So let's look at the attributes. It's important we also look at that. So let's just look at that here. So let's list the attributes. That will be the last preparatory step. Okay, so you can say attributes, and then we say list attributes. And then we specify our biomaRt objects, which is the ensemble. And then we can run it. So we can now see attributes. We have lots of them. We can use head to get just the first 6. Or let's even say first 10. Okay, so we have some attributes here. So this table here, okay, what we have here are names, description, and then the page. So with the attributes, it means that with the IDs, let's just limit it to our IDs. We can select the corresponding ensemble gen ID, ensemble transcript ID. So all this here we can get them. These are information that we can retrieve, okay, for the IDs we have. But we are going to select just a few of what we have here. You can also search, because we have lots of attributes. You can also use a pattern search. So for our data sets, yeah, this is the ensemble IDs, okay. So with the IDs that we have, we are going to convert them to gen symbols. We are going to use the Hugo nomenclature. Let's just check it out. I'll just open it here. So I'll use this to explain. So let's just search. With the attributes, you can search. So because we are going to use the AGNC, which is the Hugo nomenclature, let's just search for it first. So you can say search attributes, just as we did with the data sets. We can also search using some patterns. So you can say search mart equals ensemble, and then pattern equals, let's use AGNC, and then let's search. Okay, so that's the AGNC. Let's check it here. Okay, so that's what we have here, the AGNC symbol. So what we are going to do is to convert our ensemble IDs to this symbol here. So take note of that. With the attributes, you can also get the ensemble gen ID. We are already using the ensemble gen IDs. Okay, but it's also here. So, I mean, these are things you just have to search and then select the ones that you want. So, I'm only using this tutorial to introduce you to this R package so that you can take it up from there. So, let's go. So, we will use this one here. Okay, so now we have everything we need. So it's now time for the main work. What we did was just an intro. So we are now going to start the work itself. So let's proceed. Okay, so I'll begin again. So what we are going to do is to convert some IDs into gen symbols. Let's look at these IDs. Okay, so let's proceed. I have these IDs here that we are going to use. We will look at the first 4. So let's go to our R. So if we go back to what we did, the first step is to get our biomaRt objects. And so what do? We say ensemble, and then we say what, use ensemble, and then we specify what, biomaRt equals gens, and then data sets equals what? Homo sapiens underscore gene ensemble. This is the data set we are going to use. We can also specify a version, so we can say version equals 106. So this is what I was talking about. You can specify a version here. Let's correct the typo. Yeah, so this is the version. You can specify it here. Perfect. Let's just check to make sure that everything is okay here. Everything is fine, so we can proceed. So let's run this to create our biomaRt objects. Perfect. So we have it created. The next thing to do is to call the getBM function. So this is what we are going to do. We are going to say results. We assign the whole operation to a variable. So we say results, and we say getBM. And then we say mart equals what? Ensemble. The mart should be lowercase, so mart equals ensemble. And then we say attributes equals, because we want just one attribute, which is AGNC symbol. You can just use it this way, AGNC symbol. And then let's specify filters. So filters also helps us to restrict the queries. Okay, and so with this particular filter we use, it will help us to restrict the query so that only information related to the values that we will specify will be returned to us. So that's what we will do. So we will say, so I'll say filters equals, and then I'll specify ensemble gen ID. That's the filters. We are using just one. After that, I will specify my values. I will use only one ID, so I'll say values equals, just copy and paste here. Perfect. So I now have my query. So I'll run this command. Perfect. So everything has been done. So let's check the results. So we say results. Perfect. So we now have what our symbol returned to us. So this ID has been converted to what? HGNC symbol. Okay. Now with the values, we can add more. Okay. So let's just say we want to get the gene symbols for 4 IDs. We can specify them here. So let's just proceed. So let's just rewrite this code. Here the values, I'll use 4 IDs. So let's check. So I'll say values equals, and this time because we will specify 4 values, I have to use vector. So I'll say C to brackets, and then I'll specify the IDs. So this is the first one. Let's add a second ID. I will leave all these IDs in the description box. Let's add a third ID. Let's add a fourth ID. Okay, so we have it this way. So let's run this command. Okay, so we have the query also done. So let's check it out. Results. Perfect. So we have them here. Okay, so now we have the IDs and we have their corresponding symbols here. Okay, so the symbols here are returned in the same order as you specified here. But then we can also modify this code. So we can modify it so that we have both the IDs and then the symbols also returned to us. Okay, so that's what we have. So we will do that. So I have a script, I have an R script that will help me to do that. So with the R script, it also allows me to do this. It allows me to process lots of IDs like this one here. So the R script is available on my Patreon platform. I will leave the link in the description where you can go and then download that script together with this gen IDs here, this list here. So you can go check it out. So I'll run the scripts to let you know how that is also used to get the gen IDs as well as their corresponding or their respective gen symbols also returned for us. So let's check it out. I'll do that here. So this is the script and these are my IDs which I have opened. So this is how the script works. I'll open my terminal here and then I'll call the scripts. So I'll say R scripts and then I call the script itself, the file name. I have to specify it and I'll also specify the IDs. Okay, so this IDs is a text file that has the ensemble IDs. If you also have your ensemble IDs, you can just specify that here and then you just run it like this. Again, you may have the warning messages come, but you can ignore them. So once all the gen symbols have been retrieved, you have this text appearing. IDs have been converted. And so you can check. A file will be created. A CSV file will be created. And this CSV file will have both the IDs as well as the gen symbols. So let's open it. Perfect. So this is how the results will be returned to you when you use that R script. So you have your ensemble gen ID and then the AGNC symbol. So we have it this way. Again, the scripts, the R script is available on my Patreon platform, so you can go and download it. So that's it. And so when you have the gen symbols like this, you can now use them in place of your gen IDs. That is more convenience. The gen IDs are long. In situations like gen expression analysis, you may have your gens being returned to you as this, okay, in this format. And so using the biomaRt package, you'll be able to convert all those IDs to symbols and you can use the symbols in place of this. And this will help you to also get your reports done in comprehensive manner. Okay, so it's important. These IDs sometimes may make it difficult for your readers to understand, okay, the results of a report, but if you have them this way, it's kind of simplifies everything for you. So, and that's how it is. So that's all for this tutorial. Let me also get your comments. I'll be happy to get feedbacks on how to improve these tutorials that I make. So just put all your suggestions and comments in the comment box and then we can have a discussion. Okay, so that's all for this tutorial. So I'll see you in the next session. Goodbye.